Hi everybody, welcome to another Chats with Children. Today I'm delighted to be joined by Aaron Sato, who is the Chief Scientific Officer for Twist Biosciences. Hi Aaron, how are you?
Great, how are you?
I'm very well, thank you. So I know you've had a long day. Your day started at 5 this morning, I understand, doing lots of other virtual shows. So,
Yeah, well,
and it's now 9 p.m. in the UK. So anyway, I appreciate you taking the time out to have a catch up with me. So what I want to do is find out a bit more
thank
about,
you.
well, great. I wanted to find out about synthetic libraries. That's what we're going to talk about shortly. But before we do that,
Sure.
I thought it'd be useful if people could find out a little bit more about Twist Biosciences and your role there.
Great. So again, my name is Sato. I'm the CSO of the biopharma vertical here at Twist Bioscience. Again, I was hired about 2 years ago to come on board to create this new vertical in the company. Twist, as you may know, is a company focused on using our DNA synthesis platform to make very large alga pools, and we use those alga pools to create all kinds of fantastic custom DNA products, from custom clonal genes to NGS enrichment kits to high quality synthetic DNA libraries. And so what the Twist Biopharma vertical does is we utilize those libraries to generate our own proprietary antibody phage display libraries, as well as a whole platform for antibody optimization. And so we go out and broadly license those libraries, as well as do partnerships and projects for biotech and pharma companies.
Fantastic. Okay, well, the last time I actually caught up with Twist Live was way back at PEGS Europe, actually, way back in November in Portugal, where I did a poster video with one of your colleagues. And now a lot has changed since then. I mean, COVID's obviously hit the world. And how has COVID affected you as a business, both internally and actually dealing with your customers?
Great question. So certainly, there's lots of active COVID research going on right now, and particularly in the life science industry, and that requires a lot of DNA to be made. So Twist is actively making DNA for all kinds of different researchers to make different antigens associated with the virus, as well as helping to make antibodies associated against the virus. So we've been actively working this whole time to make sure that we continue to provide DNA to the life science industry. And so we continue to ship out all over the world with all those DNAs.
All right, brilliant.
In terms of the Twist Biopharma vertical, oh, go ahead.
No, go on. You said about the Twist Biopharma vertical. Please carry on.
Oh, within the Twist Biopharma vertical, we're actively also, again, continue on with our current projects and really driving forward to help our partners discover antibodies to all kinds of different targets and different diseases. But we also have been undertaking a large project to find antibodies to the virus, both for therapeutics and diagnostic applications. And then actually quite recently, just today, we actually launched a whole vial catalog IgG product associated with those efforts. So a lot of great work in the COVID space, coming up a lot of great antibodies derived from our own libraries to help in terms of tools, as well as to actually combat the virus.
Fantastic. All right. Well, look, let's start talking about synthetic libraries. So what makes Twist libraries different from other synthetic libraries, and why do you think they are actually better?
So again, a typical synthetic library uses a degenerate oligo to encode for the diversities in the CDR loops that encode for an antibody. As I mentioned, Twist's platform is basically a fantastic system to make oligo pools. And so we use oligo pools rather than degenerate oligos to make our libraries. That gives us a lot tighter control over the sequences that go into those loops, and also gives us a higher quality product in the end. So there's less errors and unwanted mutations, and we can really precisely create a library that matches up with our own design. So, and then on top of that, we can very, very quickly generate those libraries. So using oligo pools, it's actually much, much easier to generate a library than compared to using a degenerate oligo. So that gives us a much higher quality product to then build a whole technology platform on.
Right, okay. So have you done any work with any other display platforms that work in concert with the library of libraries in phage display?
Yeah, great question. So yeah, we're actively working on that. I think there's a lot of great publications out there showing how phage display can marry really well with cell-based display. And to really take advantage of the kind of eukaryotic mammalian filter of a cell-based display platform, coupled with the high diversity of a phage display library. And so we're actively working on a system to take in outputs from several rounds of phage display panning and port that into a milling display platform so that we can get to even use flow cytometry and FACS to sort out our highest affinity antibodies.
Right, okay. And how have your VHH single domain libraries performed compared to your scFv or Fab libraries? Are they comparable?
Yeah, actually they are. I was really surprised. Again, VHH single domains are basically half the size of a traditional antibody, and so I thought we might lose half the binding affinity. But actually, what we're finding is for certain targets where there's really kind of small crevices or unique epitopes that are kind of sometimes often occluded by a larger antibody, the VHHs work great and they bind with very high affinity. So, and I think that's a testament to again the library capability we have to create very high quality, very diverse DNA libraries that encode for antibodies. And so the VHH libraries that we use, we love reusing them routinely against all kinds of different targets, and they perform almost equivalently to the antibody libraries.
Fantastic. All right. Well, look, I know you've been doing quite a lot of talks this week at various events, and you're actually doing a presentation next week at PEGS Virtual, which would have been in Boston, but obviously now it's global on the net. So what can people expect to hear from you during that presentation?
So in that presentation, I'm going to give a global overview of all the fantastic things that we can do in the Twist Biopharma vertical, which includes talking about our library of libraries and all the new phage libraries that have come out recently. I'm going to talk about our whole antibody optimization platform called Tau. And then I'm also going to roll out our new alpha product, which is our high throughput IgG product, where we can basically couple our synthesis with high throughput antibody production. All of the fantastic COVID work I just talked about in terms of finding antibodies, both from our traditional antibody libraries, but also our VHH single domain libraries as well.
Right, okay. Well, if people would like to know more about that presentation and PEGS, there will be a link above the video which you can click on. Also, I know that Twist have got a huge sort of resources on your website with posters, webinars, and various other pieces of content that people can access.
Definitely check out twistbiopharma.com. It has all kinds of flyers and webinars and all kinds of cool resources at your disposal.
Fantastic. And so if you want to know more about Twist and the different things that they can do, please check out that link. Again, it will be on the post above the video. And if you've got any questions for Aaron, then by all means, please leave them in the comments box below the video. So Aaron, I know you've had a very long day. I'm going to leave you to it now and let you at least have a bit of a break. So thank you very much for taking the time out to have a chat to me. I hope everybody who watched this found this useful. And as always, stay well and stay safe. Bye-bye.
Great. Thank you. Bye.